properties, etc. We are going to now start with the morphology. And right, so I recently heard Larry Abbott proclaim that we've entered the connectome era, the connectome age, which means we have now chunks of neural tissue that are reconstructed with serial three-dimensional electron microscopy. And that means we have the reconstructions of all the morphologies in the tissue, all of them, and at nanometer resolution partly, including also all the synapses that connect them to each other. So we are able to really look at this relationship of morphology and actually connectivity in the circuit in great detail. It also means basically that you know, if in an experiment we find something new, and we may, you know, postulate a circuit that may be underlying this new piece of evidence, we can no longer do this. We can actually go into the data and look if this circuit exists, right? So you cannot not make up things. And these kind of tissues, these kind of connectome data sets, they exist for various species. In particular, of course, for the fly, there's a complete brain of the fly. There's complete larva imaging at various developmental stages. There's a worm and a zebrafish brain and mouse. Now we talked briefly about this also on Monday. This is really worth looking into these microns data sets. The website is really eye-opening about how the circuit actually looks like. Monkey, human, everything. It's slowly coming in. And so if you believe like me that the architecture in the brain really matters for the implementation of you know, the function, then you'll see that this is an exquisite these are exquisite data sets. If you don't believe it, I want to give you just this little example here, which is the ellipsoid body in the central complex of the fly, and these are head direction cells. So the cells respond to the direction of the head of the fly, and theoreticians have postulated that there might be a ring attractor that is responsible for calculating this head direction and constraining basically this the direction into one direction. And you see the anatomy. It comes out to be actually a ring-like structure, and you can basically read out from the anatomy the underlying math, basically, right? So this situation is, for me, always reminiscent of more than 100 years ago, and we see Cajal pictures again and again in this course, where basically a lot of new staining methods were developed, and in particular, and so it was possible to really look into the brain for the first time in detail. And in particular, of course, Ramon y Cajal was able to use the Golgi stain that stains randomly a very small subset of neurons to postulate that the brain is made of, or the nervous tissue is made of single neurons that have a dendritic tree as an input and an axon as an output. And he was able to do more than that. He was able to, from simply just the anatomical representation of what he was drawing just from the anatomy, he was able to draw these little arrows to actually infer from the anatomy how the signals move in the circuit, and maybe what may be the circuit's function eventually. So it is really important, the morphology, and to me, the morphology of in particular of dendrites, which I focus on, is kind of the primary building block of what makes up the circuit. And nowadays, we don't draw the dendrites anymore on paper. We draw them in the computer. But that's not a big difference. It's still mostly done manually. So also, I don't have to go in too much detail here, because Dieter already explained a lot of this. So there's NeuroLucida, for example, where you can just go point by point, and there's a variety of similar programs to basically draw the three-dimensional structure of these dendrites and axons. We have also our own tool, which I'm not sure I recommend, but essentially there is no there's no tool that is really perfect. In fact, we discussed this yesterday, that you have to be very careful if you use anything that's semi-automated, because semi-automated reconstruction means we put in some assumption about how the dendrites look like, right? And that means you can get this wonderful reconstruction of the cells, and then lo and behold, it's exactly the statistics that you were expecting, and it's maybe because that's the statistics you put in into your automated reconstruction. So you have to be very careful. So automated, you have to be careful. A lot of stuff is still done manually. Although the EM data sets, they have to. That's tricky. Okay, and again here a lot of things have been talked about already by Dieter. And there's a neuromorpho database which is really, really great. Contains really almost all the morphologies, non-EM morphologies that have been published. There's Fiji, of course, that has tools to do these semi-automated or manual reconstructions, and a variety of other tools and databases that I put up here. And this is by no means complete. Okay, and so now basically you can have a digital representation of this tree, and again this is not very different from what Dieter mentioned, but I just want to put it again freshly into your mind. So obviously we present a tree by a set of nodes that have x, y, z coordinates and a local diameter. These are the metric information that we need, and then of course an adjacency matrix that says which is the parent of which node. Yeah, so there's the adjacency matrix, and there is a 1 if the node 2 is directly connected to node 1. So there's this is supposed to be this tree. So if node 1 is connected to node 2, then you will have a 1 here, etc. And so you can get this adjacency matrix, which basically has one value in each row. That's the parent of each node. So that's the representation. And once you have this in the computer, you can make all kinds of measures, and they're called morphometrics. For example, measure the path length or the number of branch points. Here's only a rough summary of some of those measures that you can think of. In particular, for example, you can calculate the path distance versus the Euclidean distance and these kind of things. And the crazy thing here is, even if you really think of hundreds of those morphometrics, you can, in almost no case, actually use this to classify dendrites into their respective cell types. Yeah, so these morphometrics are not very practical for this. So for example, if you look at the principal component here of these of a large variety of morphometrics, you can see that you cannot distinguish them. What you can do is you can compare real dendrites with the set of all possible trees, for example. So this narrows down the morphological space, for example, of dendrites. So these morphometrics are not good enough. So we actually need modeling, and in particular, since I told you I'm interested in this relationship between connectivity and morphology, we need some kind of a model that takes into account these two things, right? And so the thing that we took advantage of is basically going back to Cajal, and he wrote these very, very thick books, two very fat volumes, 100 years ago, more than 100 years ago, in which he summarized everything that he drew on his kitchen table under the microscope. And he says, I wouldn't have been able to understand any of it if not all those branch structures had followed always exactly these three laws that he wrote down. The law of conduction time conservation, so that signals have to propagate quickly in the circuit. Law of cellular material conservation, and the circuit has to use little material as possible, and space conservation. And this Cajal had in his head while he was drawing, and this allowed him to put this directionality to say this is a dendrite, this is an axon, etc. And we simply use this to make the most simple toy model based on these on two of those rules. So in this model here, we assume that the input locations are known. Okay, these are the red dots, and in black we have let's say the summation point, the root of the dendrite. Yeah, and of course, so a dendrite has to collect these inputs, so it has to extend towards them, and it may do this efficiently. Yeah, so it may do this by minimizing the amount of material. So the second law of Cajal. And when you do this, then when you connect those red dots using the minimum amount of cable, then you use what is called the minimum spanning tree algorithm in graph theory, which is basically the shortest connection here to get to all of those points. And that's the tree here on the very left side. Yeah, and what's the problem here is that actually this is not an efficient dendrite, because if a signal comes in here on the outer right side, then the signal has to go all in a spiral before it reaches the center. So this is extremely inefficient for signal conduction. Yeah. And so basically, in order to reproduce dendritic trees, we had to add a second cost in top of this cable length cost, which is the path length cost, or the conduction time cost, the first law of Cajal. And that forces these paths to be as short as possible here, yeah, with one parameter that balances these two costs. Is this clear? Yeah. So just as an example, if you would have this balancing factor, if that was infinitely large, only the path lengths would matter, then every one of those red inputs would be connected straight to the root. Yeah, and this is also so both the minimum spanning tree and this very high path length tree are unrealistic and are basically not observed in reality. But anything in between with different balancing factor values are possible dendrites. And so is this clear to everyone? You really have to ask questions, because this is important. Okay, so if we assume that we know these targets, if we distribute the targets appropriately, and then we use simply this one algorithm, very, very simple, it's very fast also to calculate, we are able to reproduce morphologies of a large variety of dendrite types. For example, these granule cells in the dentate gyrus that have obviously very high balancing factor. You can see that the paths are directly connected to the root. Yeah. almost. Or the Purkinje cell here in the cerebellum, you saw already quite a few, with a much lower balancing factor, or starburst amacrine cells that are also two-dimensional cells in the retina. And in particular, of course, pyramidal cells work incredibly nicely. These are layer 5 pyramidal cells in the cortex that are essentially indistinguishable from the real counterparts, yeah, the biological. Yeah. Completely fake, yeah, completely fake. So there is nothing correct here, nothing real. So these are not reconstructions, these are actual fake data. And really, I mean, we let expert electrophysiologists look at them in 3D, and they had a real hard time distinguishing them from the real ones. But of course, I always like to oversell everything that we do. So I have one more slide to maybe make this a little bit more precise. So here's what we did for these layer 5 pyramidal cells. So first of all, we had to divide the dendritic tree into an apical part and a basal part. I think I lost the point here. An apical part and a basal part, because you have to first grow the apical dendrite, and then these oblique dendrites, and then the basal dendrites. Because otherwise, if you do this in one go, then the minimum spanning tree, this algorithm would go meander around before it reaches the tip. So this is already a caveat. Second, you may see that what we get here does not look at all realistic, right? And this is what you get straight from the model. What we did is we added a little bit of jitter, and we added a little bit of diameter taper, and suddenly this thing looks really realistic. Okay. So and of course you may say, well, actually, how did you distribute the targets in the first place? Well, this is from real data. So we took the apical dendrites of real trees, we put them together into the same context, we scaled them, and then we calculated this density cloud, and then we distributed points randomly, and then rescaled them to have the same diverse scaling as in real trees. Okay, so there are still hacks, right? It's not completely straight from math, yeah. Although you can, of course, think of ways to make it much more independent of original data. For example, you can say, okay, I have parallel fibers that are these are real parallel, right? And I connect a tree that has to connect to each of those parallel fibers once, okay? And then I get immediately a Purkinje cell, because both for minimizing the cable and for minimizing the conduction time, a two-dimensional tree is efficient. Yeah, you have to stay perpendicular to the parallel fibers, and this is exactly what you see in terms of Purkinje cell morphology. Same thing you can do also for pyramidal cells. You can assume that it connects maybe 2 layers or variety of layers, etc. And so you can make this a more geometric and less dependent on the original data. Okay, so this is clear? Yeah, questions? No questions? Okay. Take advantage. Okay. So then I said I put some taper, but this is also a little bit blurry, right? So diameters. What do we do with diameters? Well, what we found was a really good guess for the local diameters was when we generated diameter profiles that optimize the current transfer from the synapse to the root. When you do this, then you try to equalize the impact from distal synapses to make them as similar as the proximal synapses that are typically otherwise stronger. And if you do this, then you tend to see these thinner tips with a higher local input resistance, which actually leads to the fact that there's more arriving at the root. And this is actually a quadratic taper that you can derive directly from the cable equation. And this is something approximately like what we see in real data, but this is not completely the final word. So now nowadays we're able actually to fit the local diameters in an entire tree based on a number of costs. For example, also protein transport considerations and etc. And so now we can refine these diameter profiles in real dendrites using basically still this kind of optimization idea, right? This is still something that is debated. Okay, good. Now, what do we do with this model? Well, the first thing that we were able to use the model for was to look at all these morphometrics and make a sense of them. Yeah, basically, there is you can calculate, of course, the relationship between morphometrics in the model. In particular, the scaling relationship. So how do these morphometrics behave when you make the tree bigger? Yeah, and so the first thing we observed was well, in real data here, these are data from Adi Mizrahi. He has adult-born periglomerular neurons in the olfactory bulb, and they migrate to the glomerulus, and then they innervate it in this spherical structure, and they become more and more complex. And so you can see that the length grows and the number of synapses also increases, right? But there's a particular scaling relationship that you can clearly see. And this scaling relationship is perfectly predicted by our model, by the minimum spanning tree. This is a very nice data set because we have this time lapse, we have the maturation of the dendrites, and we know exactly where the synapses are. That's quite a rare data set. But we observed here that actually both in the data and in the minimum spanning tree generally, there is a particular ratio between the number of branch points and the number of synapses in the case of the data, and the number of targets that we use, the input targets, these red dots that I showed you in the beginning, in the model. And this is kind of a quarter. So if you distribute n points, n red points, then a quarter become branch points, a quarter become termination points, and half of them become continuation points. Yeah? No, not a question. Okay. So now we are able to replace this n, the number of synapses in this particular scaling relationship, by number of branch point with a particular factor with this basically 4 times, yeah, that we found here. And now we are able to put all the data from the neuromorpho database, all the dendrites that have been reconstructed, and look total length versus branch points. And there is this interesting 2/3 power that is predicted again by the minimum spanning tree for 3-dimensional trees, and a half power that's predicted by planar dendrites. And again, you can see this is a very nice scaling relationship, and the model reproduces it perfectly. Now, again, I'm really completely, totally overselling this, right? As you can imagine. So obviously, if you look at this is a log-log plot. Yeah, so it's actually a power law, and so basically these data here, they're almost 10 times the prediction from the model. But the trend, I mean, this is this very nice thing. So we actually use this scaling relationship as a signature in the morphology to say, you know, these dendrites are or are not optimally wired, and indeed in these real cases they almost always follow this scaling relationship. Is this all still clear? Still clear? No question? Serious? Okay. Okay. So we found by further investigation, we found actually a new morphometric using the model that allowed us to infer the balancing factor, the one model parameter, the one parameter. We are able to get it directly from the morphology if we assume, if you calculate this root angle, which is basically the angle from each segment in the dendrite here, of the vector compared to the vector that points straight to the root. Okay, so this angle, this root angle, if you now look at the distribution of these root angles, then you can see that different cell types have different types of distributions, and from these distributions you can directly infer the balancing factor from as the one parameter from in our model. Okay, so I mean, this should be clear. Again, this granule cell here has a very high balancing factor, which means that that almost all the segments show almost directly towards the root. So you have a very tight distribution, very small values of this root angle, right? So that's the idea behind this. Okay. Still clear, everything good, wonderful. and we also looked at the distribution of these targets. So what if we make the targets more or less regularly distributed as compared to a Poisson distribution? This is Poisson. Then you can make more distributed or less distributed. And what we find in the model, and then of course also in the real dendrites, is that you can read out from let's see, this regularity of value, the R of I think the termination points, you can read out the regularity of the inputs. So you can actually infer what is the regularity of the distribution of these red target points, and we singled out in this paper here these 4 DA neurons in the fly, which had clearly different R values. Yeah, so I said it wrong. It was about the branch points are. But anyway, so you can see that this class 3 has very clustered, obviously clustered branch points, and therefore predicted input as well. Whereas these class 4 have much higher regularity of the branch points, and therefore probably the inputs. And so we are now going to look at these 4 classes of dendrites, which are actually incidentally the model system that you use to look at dendrite development and the genetics of dendrite growth. And so depending on how much time I have, how much time do I have actually? 15 minutes. Okay. So now these are more newer stuff. So I'll go a little bit quicker since you understand everything very quickly anyway. I think that should be fine. So we're starting here with so these are the different DA neurons. They are tiling the membrane, the outer skin of the larva, and there's a class 4 neurons that are thought from experimentals thought to be space filling, and we'll go to class 1 and class 3. And so what we do here is we're actually interested in how does biology implement these optimal wiring constraints, or this minimum spanning tree. How does this happen? And so what we did in this collaboration with the lab of Gaia Tavozanis and all the PhD students involved here, we looked at the same neighboring dendrites at consecutive developmental stages. And this is time-lapse, so these are exactly the same dendrites first in the embryo, then in the larva instar 1, larva instar 2, and larva instar 3. And the nice thing when you reconstruct 2 of those dendrites here is that you can recognize from one time point to the next, you can recognize the branches. Yeah? And so now the question is, what happens during this growth process, right? Obviously, the larva becomes larger, so there's some kind of a stretching that happens. But obviously, also there are more branches that are grown additionally. Yeah, and to make it a little bit short here, at first, if you look at simply the data. Well, many of those branching statistics actually from one time point, the colors correspond here to the developmental stages. If you normalize with the size, then the branching statistics are basically identical. So there's some kind of self-similarity from one time point to the next. Yeah, and this is almost perfectly reproduced by our model. So if we do this individually for each stage here, developmental stage, then we're able to reproduce the statistics perfectly with our minimum spanning tree model with a particular balancing factor that does this very well. Okay. And so this means, since we're able to reproduce this with the minimum spanning tree algorithm, that means that throughout development, these dendrites are optimized for cable and for path links. Right? So at each time point throughout the development, while branches are added newly, we're always optimal in the sense of these constraints. Roughly, of course. Now we have another measure here, which we introduced because these cells are supposed to be space filling, and we're saying how far do I have to go away from the dendrite to fill 90% of the surface area that the dendrite innervates, and this we call this theta, and this theta is also very nicely reproduced by our simple model. Okay, but I mean, we have time-lapse data, so we can do much more than just look at the different stages. So what we did is actually, and this is mostly manual again, we said, okay, we can identify this termination point that's the same, so we call it in same color here, etc. We can, and you can see all the branch points and termination points that are conserved. You can find them in the next larva instar stage here compared to this one. Okay. Roughly. I hope you can see this. Of course, there are more branches here, but there are almost none that disappeared. So this is a bit of a special case as well. There's almost no pruning here. And so when we just look at these vectors from the conserved branches, what happens is essentially an isometric stretching. So the dendrite just becomes larger. Yeah, the amplitude of the vector corresponds to the distance from the root, so it's a linear stretching. Okay, and now what we can do, we can remove this stretching, and we can only look at the new branches. So this is the data here, and you can see in purple are the new branches when we have removed the stretching. Okay. And now what we observe is that actually the branches tend to grow towards the space that is as far as possible from the dendrite, from existing dendrites, to further space fill the available space. Yeah. And when we reproduce this exactly in the model with a bit of noise, so this has to be calibrated, then we are able to basically reproduce the scaling behavior that we know from the real cells using this one iteration of growth, and we're able to grow, of course, dendrites de novo. This is complete fake dendrite that is grown where the development is now also taken into account, and at each step, again, because we're following the scaling law, at each step these dendrites are optimally wired in terms of this signature scaling, yeah, and this iteration of growth guarantees, by being a minimum spanning tree kind of algorithm, guarantees also this optimal space filling. Yes, we get a question. Yeah.
I forgot this one. Let's try with that. Okay, like we know that if we cut.
Yeah, I can.
Yes, but it's really hard for me.
I can repeat the question.
Okay, yeah. And like if we cut, like have you tried to cut it, like and try to see the difference, you know, like if they are growing, because if you cut
to cut a piece of dendrite.
Yes,
here?
yes.
Yeah. So this has been done experimentally. So with a laser you can ablate a subtree, and in exactly those cells actually. And we have used our minimum spanning tree to repair. And the crazy thing that happens is there's two modes of regrowth in real dendrites. One is we grow back from the cut branch. The other is, so it's a bimodal distribution. We grow back from neighboring, this is basically innovated from neighboring branches. And we observe exactly the same bimodal distribution using our algorithm as well.
Okay,
so,
thank you very much.
So yeah.
I would like to add to that just the opposite direction. So layer 4 star pyramidal cells tend to retrieve and lose their apical dendrites, or reduce it at least dramatically.
Yes. Yeah.
Can you also kind of model that? Because they may lose some of the input zone in the upper layers, so that they, well, structurally retrieve this dendrite? Yeah,
so now you're talking about maybe activity-dendritic pruning or something like that. So
it's an active process actually from the cells themselves.
Yeah.
They really retrieve their dendrite.
Yeah, so maybe I show you a few more examples here, and then we can go back to this. And I also have to be careful that I stay approximately in time, 5 minutes. Okay. So you see, we can reproduce. These are fake larvae that grow where every dendrite of these class 4 dendrites is reproduced. Okay, but we can use this one iteration of growth that we got from fly larva dendrites to reproduce the developmental growth of other cells. Yeah, granule cells in the dentate iris, or other fly cells, these tangential cells. And so this is now getting a bit surprising, but again, I'm overselling, of course. And now we have to go into the other class 1 and class 3 briefly, just so that we relativize what we found here. So obviously, the growth process here in these class 4 guarantees optimal wiring throughout the development, and the optimal wiring itself guarantees that there is an optimal space filling. And it seems to be a useful basis, at least an underlying growth program for not only fly dendrites, let's say. Not only class 4 fly dendrites. And so then we went on, and I will have to go relatively quickly over this, but you may also ask me afterwards. So now we have these class 1 dendrites, and they have this comb-like structure with so this is data from Andre, who was a PhD student in my lab, and he actually imaged the calcium signal during the contraction of the larva, and these dendrites by having this perpendicular shape, they are ideally suited to sense the contraction of the larva. And what he found when he did this time lapse is, in this particular case, everything happens in the embryo. Afterwards, the scaling is just a stretching. And there is an outgrowth, but then there is a pruning. And this pruning seems to be dependent on the function. And I have to keep it short, but basically, so you can see this, of course, there's these stages, so obviously different programs that happen. And throughout, even though there is a pruning, even then we remain on the scaling law to be optimally wired at all times. And so the big question was, how does this pruning happen? Is it specifically branches that have a bad angle that get pruned to keep these perpendicular branches? And it turns out, I skipped this, but in a model that reproduces the data really very nicely, all Andre had to do was to grow out branches with a class 4 program and then prune branches randomly. And by this kind of random outgrowth, random pruning, he magically got this more or less this comb-like structure and exactly the angle distribution that you get in the real dendrite. So this is the exact function of this. You have to think a little bit about why this is the case. But what I really want to make sure that you take home here from this is there is obviously a second program, right? There's an outgrowth program and there is some pruning program, and these dendrites, by the temporal sequence of these programs, manage to generate the shape that is ideal for the function in this particular case. So that's what I mentioned here. And then finally, we also looked at these, just to give you another example, these class 3 dendrites that, that have these clustered branch points. Yeah, this was the case that you can see this, right? This looks like no other dendrite you've seen probably before. And if you model this with the class 4 kind of outgrowth, yeah, then you get a completely wrong space filling, right? And so here we have to implement a two-step growth model. One, where the primary branches are grown with a class 4, and one with a class 4 program, and a second program that is completely different from the others, that is specific for the small terminal branches. Okay, and this also means that the scaling law here is perturbed. This is not doesn't follow anymore the space filling and minimum spanning tree growth. And this also allows us now to find out what the specific mutants or specific genes what role they may play. Okay, so basically what I want to have here as a take home is that there is some core program of outgrowth. And there is then individually for the different cell types probably different sequences, different programs that refine the structure. And so it's probably much more complex than we think. And we're now able to use this growth program also in a space packing way, so to reproduce actual connectome data. Yeah? And this is going to be used also in the next talk by Peter for looking at this structure-function relationship, and of course in other complementary models of dendrite outgrowth. We have also the trees toolbox, which is a MATLAB tool that allows you to do all the modeling, but also the reconstructions, measurements, and morphometrics and everything. We're thinking about going to Python, but this is a big thing. It's hundreds of thousands of lines of code. It's also an interface with Neuron and interface with Neuromorpho. And yeah, these are the people who specifically worked on the data that I showed.
Thank you very much. Do we have any questions? Right, so let's start from here.
Yes, so if I understood correctly, you look at it as an optimal wiring problem. So there are some points and this neuron grows
Right.
out. I was wondering, how exactly does the neuron know that it grows to these points? Does it have some sort of global rule or programmed maybe? Or
So this is very phenomenological, and that's actually a weakness of the original model that we had to really put in the points, and the points were kind of inferred from the real data. In the second part here, when I talk about the dendrite growth, of course, this gets refined. You don't have to tell the dendrite anymore what are the targets where you have to grow to, but it's more like an innovation area. And so this is something you may be able to implement with some kind of molecular gradients or something like that. But we haven't made this link between the molecular mechanisms yet and the growth. So it's we hope to merge with, as I said, with other models that don't look at the function so much, but more at the actual growth process. And so this has to merge. These two have to come together, or more than 2.
Still very cool work from basic principles, sort of.
Yeah, thank you. Yeah. The optimization
Thank
works.
you. And
Okay. Yeah, so this is something that the genetic background, I cannot say much about it yet. Yeah, so obviously in specific cases we are able to tamper with particularly with these DA neurons, because they're the whole genetic toolkit is there, so you can look at all kinds of mutations. And one thing that we've been looking at specifically is, of course, cases where the optimization criteria don't hold anymore. There are some very rare cases, like the class 3 dendrites that I showed you here, and so that's of course useful to identify, cases where you deviate from this. But we haven't found, we haven't dissected this genetically. I mean, I cannot do this anyway, so this is only in collaboration.
Maybe you can imagine what would happen if this mechanism stopped working. What would be the functional consequences for the brain? Would it be slower? Like, I don't know, it would develop wrong? Yeah,
I mean, that is hard to say. So basically, there's from the optimal wiring kind of considerations, you can also make conclusions about. That's the 2 slides I skipped here about the organization of the brain. So for example, neurons that are connected to each other should also be located near each other, and so this explains also layering and topography and things like that. And so there are some mutations, as far as I know, that where, for example, the lamination of the cortex is hampered, and the animals still survive, but it's just less efficient. And so, yeah.
Okay. So thank you very much. Unfortunately, we don't have time for more questions. So
thank you.
Thank you once again for the lecture.